Hello and welcome. In today's video, what we're going to be discussing is the cell cycle and mitosis, or how your body, specifically your somatic or body cells, create copies. So we'll be going through both the cell cycle as well as the different stages. We'll be hitting each stage individually and describing a little bit about it. So, without further ado, let's get started. So cells, remember, they multiply by dividing. So cells divide for repair, growth, and reproduction, with repair and growth primarily dealing with mitosis, and reproduction primarily dealing with meiosis, which we'll get to in the next video. So in eukaryotes, cells divide via mitosis and meiosis. Mitosis for your body cells, and meiosis for your gametes or sex cells. In prokaryotes, however, they divide via binary fission. In binary fission, what they do is they go ahead, just double all the information, and then cut themselves in half. Now, we'll go ahead and describe the distinction between mitosis and binary fission here in a moment, but they are very similar. So the cell cycle for somatic cells, remember somatic is body, so it has two main parts. It has interphase, which is the life and growth phase where the cell is just kind of hanging out, being a cell, and getting ready to divide. After that, we have mitosis, which is the division of the nucleus, and this is what goes ahead and differentiates the two, of binary fission and mitosis. We'll also, just for the record, be considering cytokinesis, right here, as the end stage of mitosis, which we'll talk about here in a bit. So, interphase and the different phases of mitosis. So interphase is the phase where the cell is normally in a living phase. So the cell's just kind of hanging out. In this case, DNA is called chromatin. So the chromatin, right here, as you can see here, it's these kind of thin, invisible, stringy substances. Now this is your DNA before it's actually condensed into your chromosomes. So during the G1 phase, or the first gap phase, the cell is just living, doing normal processes. During the S phase, or synthesis phase, the DNA goes ahead and replicates, and makes a copy. Finally, in the G2, or the growth stage, the cell is expanding, and replicating all of its organelles, getting ready for division. It's important that it goes through with the S phase and G2, the synthesis and growth, to make sure that the resulting cell, when the cell splits, has both all of the organelles and genetic information that it needs. The last thing you'd want is to go ahead and go through and make a copy of a cell that's missing something like a mitochondria. That could lead to some very, very large complications later on. So after interphase, we go through mitosis. So, just a reminder, mitosis, the distinction here, is the division of the nucleus, whereas cytokinesis is the division of the cytoplasm. So mitosis is division of nucleus, cytokinesis, division of cytoplasm. So in mitosis, diploid somatic cells divide to become diploid somatic daughter cells. So remember, diploid, we're talking 46 chromosomes, and somatic, we're referring to body cells. So, the easiest way to remember this is mitosis makes an exact copy of the original cell. So if the original cell was diploid, 46 chromosomes, then that means that the daughter cells will also contain 46 chromosomes. So the stages of mitosis are prophase, metaphase, anaphase, and telophase with cytokinesis. So we have prophase, metaphase, anaphase, telophase, and finally cytokinesis. Now we'll be discussing each of these a little bit more in depth on the following slides. So in prophase, what you'll notice is the chromatin, that kind of thin, stringy, invisible substance that our DNA was, goes ahead and starts to thicken up into our chromosomes, which are denoted by these X's. From here, the nuclear envelope, or the nucleus, starts to go ahead and disintegrate, and you can see that it's no longer one continuous circle, it's starting to break up. Also, our centrioles go ahead and start to migrate to opposite poles. So this will end up here, and this will end up here. What those are eventually going to do are going to help pull our chromosomes apart and ultimately lead to our two new cells forming. So those are our centrioles. And the centrioles will start to produce what are called spindle fibers. Spindle fibers are these kind of little strings up here, which are going to be used to help pull our chromosomes apart. During metaphase, the spindle fibers go ahead and attach to the center of our chromosomes. So the chromosomes line up along the middle of the metaphase plate, or the middle of the cell. So a way to see this, metaphase, remember middle. So the chromosomes line up, and the spindle fibers then go ahead and attach themselves to the centromere of the chromosome. So metaphase, middle. And now our spindle fibers have attached to the chromosomes themselves. In our next phase, anaphase, the chromosomes are pulled apart. So as you can see, the spindle fibers have now gone ahead and separated our two chromosomes, our chromosomes back into regular chromatids. So the sister chromatids are pulled apart to opposite poles. Now this is important because when they're pulled apart, this means that each side will get the correct information they need. During telophase and cytokinesis, so with telophase, our nucleus starts to go ahead and reform. So as you can see, the nucleus is starting to go ahead and reform itself on both sides of the cell. And the chromosomes will then relax back into chromatin. So they'll go from these nice and condensed forms kind of back more into these squiggles that you'll be seeing here. So they return back to the G1 phase. After that, cytokinesis then occurs. So cytokinesis is, as you can see here, the cells are already starting to pull apart. But when cytokinesis goes into effect, it will separate both cells completely, so we will then have two separate cells, each one being identical. So cytokinesis. In animal cells, it goes ahead and it gradually starts to pinch it in half. Now this part where the two start to go ahead and pinch is called a cleavage furrow. This occurs because there's no cell wall, so it's kind of like just pinching an egg in half, except, you actually get it in half and you don't have it explode all over yourself because, eggs. Whereas here, in plant cells, what ends up happening is the formation of something called a cell plate. So a new cell wall will eventually go ahead and grow between these two cells. Considering that it has the cell wall normally, you can't really pinch it in half. So the cell plate will eventually form a new cell wall. This right here is a nice summary of everything we just discussed. So this will go ahead and tell you all about, well, this video would have shown you a little bit about what mitosis is, as well as the process and what happens during each stage. Now it's important that you understand prophase, metaphase, anaphase, telophase, cytokinesis, as well as interphase. So don't forget, during interphase you have the G1, the S, and the G2 phase. You will have to be responsible for noting what each of these phases are and what happens during each phase. So, with that, if you have any questions, please go ahead and be sure to talk to your instructor. Other than that, thank you very much for your time, and take care of yourself. I will see you in the next video.